Good day everyone, my name is Senator Idris Erinlakatabu. Today we are looking at a topic, the structure of human eye. You see, when we're talking about the eye, we all know that eye is one of the sense organs in the body and it's always recognized as an organ of sight. And it is kind of a very vital organ, or let's say one of the delicate organs in the body. The mammalian eye or human eye lies in the depression of the skull called orbits or eye sockets. The eye is held in place by 6 rectus muscles within this depression called the eye sockets or orbits. And these 6 rectus muscles, they are the ones that allow for the rotation of the eye without moving the body or without moving the head. The rotation of the eye is being controlled by one of the cranial nerves in the body called oculomotor nerve. Now, the 6 rectus muscles are these. What are these 6 rectus muscles? We have the superior, we have superior oblique. To look at the 6 rectus muscles, 6 rectus muscles that hold the what? That hold the eye in place. We have the superior oblique, we have the inferior oblique. That is 2. We also have a superior rectus. We have superior rectus, and we also have an inferior rectus. Then the last 2 are lateral recti. We have lateral recti. When we say recti, that is the plural for rectus. These are the 6. This one are 2. This one is 1, making 6. Now, this is the 6 rectus muscle that hold our eyeball in place and also allow for the rotation of the eyeball without moving the head. Now, inflammation of any of these rectus muscles, let's say some of them, can result to a condition where an individual, or let's say where the eyeball of an individual will be looking at opposite direction of a single object. This is what I'm trying to say. The case is what we refer to as we call it strabismus. That is strabismus. That is when the 2 eyeballs are looking at the direct opposites of a particular object. For instance, now we can say, let's say this is a particular eyeball, this is a particular eyeball. So when they are trying to focus on this particular object, a particular eyeball can stay here, while the other one will stay here. Instead of them to even come together so as to look at a particular object at a time. So in this case now, this is what we call misalignment. We call it misalignment of the eyeball. So misalignment of the eyeball is what we refer to as strabismus. Now in this case now, if the condition is somehow severe, we can have a situation where this particular eyeball will be focusing on this particular eyeball will also be focusing on this, and the 2 objects will be produced at the same time because the brain will now find a means to interpret each of the images, meaning that the image captured here will be interpreted, the image here too will be interpreted, and when the 2 objects are now interpreted within the brain, that is what we call a double vision, and that is what we refer to it as diplopia. That is diplopia, and diplopia is what we refer to as double vision. At the same time, in some cases as well, we can have, instead of these 2 objects to look at the 2 eyeballs to look at a particular object, and they have a particular structure or image at the back of the retina. Now, what may likely to happen is that this may not be able to focus on this properly. This will not be able to focus on this properly as well because the 2 of them are looking at that particular object in opposite direction. So in this case now, there could be a low vision known as amblyopia. We call it amblyopia. That's what we call lazy vision. We call it lazy vision. That is a lazy eye or lazy vision in some cases, as a result of strabismus or as a result of inflammation of any of these rectus muscles. Now, another point is this. Human eye, you can say human eye is more or less spherical in shape. It's more or less spherical in shape. That is when we look at the structure of a human eye, it's more or less spherical in shape, and it's about the size range. We can say the size range between 2.1 centimeter in diameter. That is 21 millimeter in diameter. To what? To 2.7 millimeter, centimeter in diameter. As I said earlier, that the highball is vital organ. So as a result of this, it has to be protected by certain structures. So let's now look at the protective structure of human eye or mammalian eye generally. Let's look at the protective structure now. To look at the protective structure, we said the protective structure of eye. The protective structure of eyes. Let's look at protective structure of eyes. What are these? Let me just see this name first. Number one, to look at the protective structure of eye, as it is being recognized as a vital organ in the body. Number one, we can talk of the eye sockets on its own. Eye socket, or we call it orbit. That is the orbit. Okay. Two, we can have conjunctiva. Conjunctiva is also a protective structure. Three, we can have eyelid. There's what we call eyelid. Four, there's what we call eyelashes. Eyelashes is another protective structure of the eye. Five, we can talk of lacrima. Even before this lacrima. Five, we can talk of four. 1, 2, 3, 4. Okay, 5. We can talk of a highbrow. Highbrow is another protective structure. Then the last one, but not the least, is the lacrima. We also have a lacrima glands. We have lacrima glands. So these are protective structure of the eye. To start with, now to look at the protective structure of the eye, let's now take it one after the other. So we have the eye sockets. I said eye sockets is a depression in the skull in which the eyeball is contained, and it is one that majorly protects the entire eyeball. It protects the entire eyeball from external injury. As you can see, the eye sockets provide a major protection for the eyeball. Number two of it, we have a conjunctiva. You see, when we're talking about the conjunctiva, we can best understand the function of the conjunctiva when we have the illustration, or let's say the diagram of the anterior view of the eyeball. Let's assume this is anterior view of the eyeball. We can say the anterior portion of the eyeball is this. Let's say the anterior portion of the eyeball. Here we can say we have the iris. At the center of the iris, we have the pupil. And here we have the sclera. This is the sclera of the eyeball. So the white part of the eyeball is what we call the sclera. The entire thing here is sclera. So here we have the iris as the disc-like structure that back at the center of the eyeball. Then we have the pupil, that is the hole, or let's say the center of the iris. Now, the entire anterior view of the eyeball is being protected by conjunctiva. So that's why we refer to conjunctiva as a thin membrane that protects the entire eyeball from external injury as well. But the major function of this conjunctiva now is to protect the eyeball from certain bacteria and dust. That is why when it gets to a level that it's accumulates, or there is accumulation of this dust or certain bacteria at this conjunctiva, the conjunctiva can easily become inflamed. And when it becomes inflamed, the condition is called conjunctivitis. We call it conjunctivitis. And when we're talking about the conjunctivitis, is what is normally or commonly known as Apollo. Some people call it Apollo. Here, the effects will not be shown in the what? It will not affect the eyeball at all. It doesn't affect the entire eyeball, as the thing will just get healed on its own. The more there is discretion of the tears, and then the fasting, the heal of this conjunctivity. Now, the third one is the eyelid. When we're talking about the eyelid, we can say eyelid is an extension of the conjunctiva. Or let's say it is a flabellar structure that forms from the extension of our conjunctiva. You can see now here we have our eyelid. This is the eyelid, and this is also the eyelid. The function of the eyelid is to protect the eyeball by what? By reducing the amount of the light that enter into the eyeball. That is why we are able to close our eyes. If you see that the kind of a large amount of light is trying to enter into your eyeball, you can easily close your eye like this. So when you close your eye, you are able to close your eyes with the help of this eyelid. And not only that, the eyelid also helps us to spread the water on the surface of the eyeball by what we call blinking. So as we blink our eye, and the process of the blinking helps us to spread the water on the surface of the eyeball, because the eyeball must always remain moist. The surface of the eyeball must not dry up. So it has to prevent the eyeball from being dried or from what we call desiccation. The third one is what we call the eyelashes. When we're talking about the eyelashes, these are the fine hairs. There are fine hairs that grow at the tip, or let's say on top of our eyelid. Here we have the eyelashes. That's our eyelashes. We have the eyelashes here as well, but we have more on top here. So the function of the eyelashes is to shed the dust out. They provide a shed for the what? They shed out the dust from the eyeball. That is a major function. And they also filter dust. They also filter dust. So when we are trying to walk in a place where there is so much of dust, we will discover that this at the tip of these the eyelashes there will be dust. So those are the dust that ought to have entered into our eyeball. So the function of this one now is the protective function of it is to filter the dust out of our eyeball. So that is the function of this. Now we also have the eyebrow. When we're talking about the eyebrow is not really part of the eyeball, but it is found on top of this eyebrow. At times it's just to decorate us. If we don't know, this is where we have the eye. We have the fine hairs here that we call eyebrow. Eyebrow, aside it serves as a protective structure of the eye. Eyebrow can also serve as a means of facial communication in some cases. So that is the function of the eyebrow as far as that is concerned. And as far as protection of the eye is concerned, the eyebrow helps us to prevent the sweat from reaching the eyeball. As the sweat is coming from our head. When sweat is coming, it will come along with certain bacteria, and as we are looking at the content of the sweat as well, it's somehow salty. So if this salty substance gets into the eyeball, they can eventually destroy or gradually destroy the cells of the eyeball. So that's why when they are coming, you get into this eyebrow, they become dry. So when they become dry, they can just be coming down. So that is the major function of the eyebrow. So that means eyebrow is not a vestigial structure. It performs its own function in the what as regard the protection for the eyebrow. So that's all about that. The last one is the lacrima. It's the lacrimal glands. We all know that our eyes produce the tears. The water, particularly when we are crying, when we are so happy, when we are so excited, water can be coming out from our eyeball. So what is the source of this eye? The source of this, what's the source of these tears? The source of the tears is what we call the lacrimal glands, that we call the tear gland. And where is it situated in our eye? So the lacrimal tears is situated here on top of this, or at the corner of the eyeball. And as you can see, this gland is one of the exocrine glands in the body. When we say exocrine gland is the gland that has a dot, that has a channel through which the substance being released by it will pass through. So here we have the tear dots. As you can see, here we have our tear dots. This is the tear dots that it passes at beneath the what? Beneath the eyelid. At the same time here, we also have another one here that passes through this place. So when the tear is being produced, they can just drop anywhere here, any open space here. But normally, they don't usually drop at this corner. So at times they get to this side before they begin to drop. So here we have the anterior end of this our conjunctiva, and this place is what we call nictitating membrane. It is formed from the third membrane of the what? The third membrane of the eyelid. Now, the tear gland can still pass through here, and as they pass through here, this is the nictitating membrane. Here we have the tip here. At the tip here is what we call nictitating membrane. That's the nictitating part of it. Now we now have the tear gland that just runs down like this. This is the tear gland, and as we have the tear gland that is turning to this place, they drain or they empty their the secretion into the what? Into the tear sac. Here we have tear sac. As you can see now, the function of the tear sac is to contain the water and empty that tear into what? Straight into our nasa, into our nose, through what we call passes through nasolacrimal ducts. That's why when you see some people crying At times, water may not even coming out from the eye. Instead of that, they will be having running nose. So what leads to that running nose? Running nose is caused by as a result of deposition of this water, or let's say deposition of the tear from this our tear sac into our nasal cavity. So that's what brings about the running nose when one is crying. So that is not the only function of the tear gland. Tear gland also contains the tear, and also contained in this tear is a digestive enzyme called lysozyme. It has digestive enzyme called lysozyme. This lysozyme is a very destructive enzyme that when it comes in contact with foreign particles that enter into the water, that enter into the eye, it is the function of this lysozyme to destroy and digest these foreign particles that enter into the eye. And with the lysozyme and the dead bacteria or any foreign particles that get into the eye, they can eventually come into the eye and form the eye pus here that we call, that normally stay here in the morning, and that is what we call slippers. We call it slippers. That's what we call slippers. That when you wake up in the morning, we normally remove it from our eye as the eye residues. We can call it the slippers. But the common name given to it, as far as the biology is concerned, we can refer to it as rim. We call it rim. So that is the function. That is another function of the tear of the lacrimal gland, to secrete the tear that bears the lysozyme that help us attack any foreign particles such as bacteria that enter into our eye. As it also collects the eye residue, the dust contained in the eye. When it collects them, with foreign particles contained in them, they form the pus here that we call the slippers, and that is the rim. So these are the structures of the word. These are the protective structure of our human, or let's say mammalian eye. Now to look at the structure of the eye, entire eye, for better understanding, or to let us know, or to teach us how to draw the human eye. So for better understanding of this now, we will look at the structure of the eye in this way. The structure of the eye has three major parts. The structure of the eye has three major parts. So let's clean this and look at the structure of the eye. Let's clean this and look at the structure of the eye. To look at the structure of the eye, I said the structure of the eye, let's say the entire eyeball, is of three layers, or is of three parts. And what are these? Number one, we can talk of sclera, or we call it sclerotic coats. It is sclerotic coats, that's number one, or sclerotic layer. That's the outermost layer. The second one is choroid. We have choroid as a middle or second layer. And while the third one is retina. The third one is retina as the innermost and the most delicate layer of the eyeball. So let's now look at this one after the other to have a distinct or well-defined structure of human or mammalian eye generally. Now to start with now, choroid or sclerotic coat. Sclerotic coat is a kind of a tissue that is made up of fibrous tissue, or structure that is made up of fibrous tissue. And we do refer to it as the white part of the eyeball. It's one that surrounds the entire eyeball. And as we are seeing it, it is not transparent part, or let's say it is not transparent part of the eyeball. Meaning that the light cannot pass through it. The light cannot pass through it. Its own function is to provide a kind of a support for entire eyeball. And it's the one that forms the outer membrane of the eyeball. Now let's now look at it now. To look at the sclera, we can say the sclera is of structure like this, and the sclera has the anterior part. The anterior part forms what we call the bulge, and that bulge is the only part that is somehow transparent in the what? That is transparent in the sclera. You can simply have this now as well as our entire sclera. And don't forget that this is the entire eyeball. The eyeball is situated in the orbit called the eye socket. Now, and I said it is held in place by what? By 6 rectus muscles. So we can simply indicate our rectus muscle like this. Let's assume these are rectus muscles that hold it in place on top. That is the superior one. We can call this one superior oblique or superior recti. Here we also have the inferior recti. Now, this is the entire eyeball now. The anterior portion of the eyeball is the transparent part of it, which is called the bulge part, is what we call the cornea. But before having our cornea, there is what we call the conjunctiva. Let's say we have the conjunctiva. Conjunctiva is one that it is a layer, and it is a membrane that surround the what? That surround the anterior portion of the eyeball. As I gave you the other time, that let's say this is the anterior portion of the eyeball like this. Here we have the sclera. This is the sclera. The entire part of the eyeball is the sclera. But the anterior view of it here, we have what we call the iris, and at the center of the iris there will be pupil. So what surrounding the entire anterior view is the conjunctiva. So to now have it side now, to have a side view of it. So this is the entire structure we are going to have. So this is what I'm trying to give out now. So here on top we are going to have our conjunctiva. Here we have our cornea, and that is the only transparent part of what? The only transparent part of our sclera. So this is the entire sclera. It is the sclera that forms the entire shape. Let's say the entire, it's one that provides support for what? For the entire eyeball. Are we getting that now? Let's now look at the second one, which is the choroid. I said choroid is the pigmented part of the eyeball. The choroid is the middle layer, and at the center is the pigmented part, as it is one that is well supplied with blood vessels. You know, the eyeball needs the oxygen and other nutrients for survival of their cell. Now, what now provides them with this oxygen and the nourishment and the nutrients is the what? Is the blood well supplied. I mean, that is the choroid, because choroid is well supplied with the blood vessels. Now, we now have what we call the pigmented layer here. That is the middle layer. We can simply indicate it like this. Let's indicate it like this from here to here. To have our choroid, we can now say it is a pigmented layer, and it's very thick layer. A very thick layer. So when we are drawing it, we indicate the very thick one. You can see, and it is somehow pigmented. It is pigmented layer. It is a pigmented layer of it. So it has pigments. And what are these pigments we're talking about? That is the melanin, majorly melanin, because it is parts that form the iris. The part of this form the iris, that is other pigmented part of the what? Part of the human eye. Now, as you can see the choroid now, choroid is the second layer. I say this is the sclera. To not forget our labelings. We say this is the sclera, and I call this one rectus muscle. Let's say rectus muscle. Here we have our choroid. This is our choroid. Now, anterior end of the choroid is now branched into two. And what are it? It's branched into iris and the ciliary body. For the iris now, iris is one that forms a disc. The disc-like parts of a eyeball is what we call the iris. And as why do we call it disc-like structure? Because it has at the center that contain hole. That hole is what we call the pupil. So here we have the iris like this. And before we can indicate the iris, let's indicate the center of the eyeball. Let's say this is the center of the eyeball like this. Here we have our iris that is coming like this as a disc structure, and this is part of, let's say this is the iris. This is another iris. So here at the center of the iris is what we call the pupil. Are we getting it now? So we can simply indicate this one as our iris. We can indicate this one too as our iris. Just pick anyone. Now, the other parts or the other components of the choroid is the ciliary body. And this ciliary body has what we call ciliary ring, ciliary muscle, and part of it is what we call suspensory ligament. And let's now indicate the three. Here we have the ciliary muscle. Here we have the ciliary muscle. And very close to ciliary muscle, attached to the ciliary muscle is the ciliary ring. And here we also have the ciliary ring. Now at the center of the two, the function of the suspensory ligament rather, we have a suspensory ligament, suspensory ligament. As we are looking at this ciliary muscle, the function of the ciliary muscle is to change the curvature of the lens, because they are the one that hold the lens in place through the suspensory ligament. Here we now have our lens. This is the lens. We can see that our lens is found at the back of the what? At the back of the iris, at the back of the pupil. This is the pupil. Here I said we have the pupil here. This is our pupil. Now this is the what? This is the lens. It indicates our lens. Let's say this is the lens. Now I said we have a suspensory ligament here. Suspensory ligament here. The function of the suspensory ligament is to change the curvature of this lens. So let's say we have the lens like this. When they contract, the lens will just reduce, and when they relax, the lens will wide open. So that is when we say we change the curvature of the lens as what we also call squint. When you have to squint your eyes as well, you can easily contract your ciliary muscle. That will help you reduce the size of your lens so that tiny amount of the light will be able to enter into your eyeball. So that is the function. Here we now have a suspensory ligament. So let's draw, we can label it anywhere. We can label it here, we can label it here. So because of space, let's use this one. We have a ciliary muscle. And here we have a suspensory ligament. Suspensory ligament that holds the lens in place. So this is all about the what? This is all about the choroid. This is all about the choroid. The third layer now we are to look into. And before we even go there, before we even go to the third layer, we can see that here, when the light enters through this cornea, it just passes through our pupil. And from the pupil, we have the lens at the back. And there is the first thing that attracts more light, that refracts more light, is this cornea. But on getting to this, before they now get into this lens, there's another thing that will refract this light onto the lens so that the light will be able to get into the lens. And what is that? There is a liquid here. The liquid is called aqueous humor. We call it aqueous humor. It is this aqueous humor that refracts the light. It's found between the lens and the what? And the iris. So that's all about that. Let's now go to the third layer. The third layer and the most delicate layer is what? It is the retina. Why is it called most delicate layer? Because it is on which the image is being formed. The image formed on the retina. If the image or light enter into the eyeball and it does not reach the retina, no image will be formed or interpreted in the brain. That means such a person will not be able to see at all. But when the image gets onto the retina, that's where the image will now form. And it will now get into what? Into our brain. Let's now look at this third layer now. The third layer is what we call it retina. We call it retina. That is the retina. Let's indicate our retina here. Now, when we say the image form on the retina, what really happens is that the retina has what we call photoreceptor. There are photoreceptors present in the retina that even make the retina to be a retina or to be a structure on which the image is being formed. And what are these things? What are these photoreceptors? There are two types of photoreceptors found or contained in the retina. And what are these? Here we have rod and we have cone. To look at the two cells, they are cells on their own. To start with, when we're talking about the rod, it is one that is very sensitive to the dim light. Why is it sensitive to the dim light? Is because of the fact that it has a kind of a visual purple. And the visual purple present in it is what we call rhodopsin. That is the visual purple of it. And this visual purple is usually what? It's usually it is being produced by what we call retina. There's what we call retina contained in this visual purple. And that retina can be nourished by, can be stimulated to produce by what we call retinol, called vitamin A. That's why lack of vitamin A can be simply referred to as night blindness, as if one will not be able to see well in the dim light. So that is the function of the rod. It is the rod that only indicates between black and white. And what is that? It's used to see in the dim light or in the night. That is what we call the rod. But on the issue of the cone, also contained in the retina, the cone is one that, even according to some facts, there are up to 125 millions of cone contained in the what? Contained in the retina. And as you can see, the cone is one that is very sensitive to colors. It is sensitive to what we call spectrum, because there are different types of pigments present in the what? Present in the cone. And cone is of three types as well. We have the red cone, we have the blue cone, and we also have the green cone. As each of these cones has what we call is a kind of a specialized pigments on their own. Now, it is the cone also have a visual purple that is called iodopsin. The visual purple of cone is what we call iodopsin. That is just that. We call it iodopsin. It's one that is able to interpret what we call spectrum. And what common example of spectrum like rainbow to identify different colors. When we are to identify colors like blue, green, yellow, white, purple, and others, so with the help of this, white is not there. With the help of this iodopsin that contain or that form the cone, we are able to what? We are able to differentiate the color. And at the same time, there is a point in this retina where it is being concentrated with what? With cone. And that particular place is what we call it foveal centrally. You can simply indicate that here. We can indicate that here as well as our foveal centrally. We call it foveal. You can just say foveal, or you write it as foveal centralis. And this foveal centralis is what we call yellow spots. We call it yellow spots. And anywhere that is being concentrated by this foveal is where the image will be clearly seen. So image that is clearly seen, that means that is where the focus, when the light, when the foveal focus on that, that is where the image will easily be seen clearly under that foveal. Now, after the image has now been formed, they turn to an impulse, and this impulse has to travel to the brain, because we shouldn't forget that I said eye is what? It's a sense organ. It's a sense organ in the sense that it's 100% depend on the what? On the brain. So if not because of brain, the eye will just be say, we just be there, we get the image, but nothing for you to interpret the image for it. And as a result of this, there is a point where there is a point now that the image, or let's say the visual purple, will meet with the optic nerve that will carry this impulse into the brain, and at that particular place there is no image formed. Nothing is being formed there. There is no vision. There is nothing that will occur there, and that is why that particular place is what we call it a blind spot. We call it blind spot. So let's now indicate that. To now indicate that, now we just have to remove certain parts in this, because we are going to indicate our what? Our optic nerve. Optic nerve is one of the cranial nerves that is sensitive to image. For us to be able to see image, for us to be able to identify images, so we have what we call the optic nerve that carry the image form at the back of the retina straight to our brain. So let's now indicate from the retina here. Here we can have our blind spot. So let's say this is a blind spot. Nothing happened. So after that, we now have the continuation of this, the continuation of this, the continuation of this, the continuation of this, and the continuation of this. All these things is one that now goes straight to the brain. They are one that carry impulse to the brain, and that is what we call optic nerve. We call it optic nerve. So this is the structure of the human eye. And before that, the image has to be refracted onto the what? Onto the retina, and that is exactly the function of the liquid that is found between the lens and the what? And the retina. And that liquid is called what we call vitreous humor. So here we have what we call vitreous humor. We have vitreous humor. So that is the liquid that refracts the light from the lens onto what? Onto the retina. This is a simple structure of what? This is a simple structure of human eye. Now, before we go on that, we can simply now look at the eye defects. Let's look at some eye defects and their correction. For instance now, let's assume the image just enter into the eyeball like this. As it gets to the lens, it passes through the lens, but instead of it to get into the what? Instead of it to get into the retina here, it does not get into the retina, it just stops here. In this kind of situation now, when the image that the light that enter into the eyeball rather, does not reach where the image will be interpreted. This case now is what we call short-sightedness, that we call myopia. This is myopia. To look at eye defect, the first one that we can talk of, myopia. We have myopia. Myopia is what we refer to as short-sightedness, as the image does not reach where the light does not reach where the image is going to form. That is what we call short-sightedness. And for the correction of this now, one has to use a lens called concave lens, and that concave lens is otherwise known as diverging lens, as it's going to diverge the image inward. so that the image will be able to fall on the retina, and the image will be, the light will be able to fall on the retina and the image will be formed clearly. So that is just the case of short-sightedness. And as the person will not be able to see an object that is very far to him or her, the victim will not be able to see. And for that, the function of that concave lens or divergent lens is to move the image, to make the light to form on the retina. It drags the image to it as it diverges the image inward. So it moves the image to very close to such a person. Either the person moves to the object, or it moves the person moves the object very close to him. So that is where the image will be seen, and that is exactly the function of what? Of a concave lens. So that's all about that. Number 2 of it now is what we call long-sightedness, or we call it hypermetropia. Hypermetropia. On this case now, to look at hypermetropia, that is when the lights that enter into the eyeball pass through the what? It passes through the retina. It's now formed at the back of the retina. It forms at the back of the retina. And when such a case is happening, the person will not be able to see a very close object. The person will not be able to see a very close object. So that person has to move back. So it's either the object is being moved back, or the person himself move so that the image will be able to reach this retina. Because when they move very close to each other, the image will be beyond the what? Be beyond the retina where the image is supposed to be formed. And to now correct this without even moving closer or far from the object, there is a corrective lens that we call convex lens. And convex lens is what we call converging lens. For this converging lens now, is one that will now move this thing outward, meaning that the object will move outward the focal point, and the image will just form at the back, exactly at the back of the retina. So that is the correction for what? That is the correction for long-sightedness or hypermetropia. The third one now that we can talk about is sightedness. We call it old-sightedness or presbyopia. We call it presbyopia. This is a condition where the image is unable to form clearly on the back, or let's say on the retina, as a result of the weakness of this one, the weakness of the ciliary muscle that's supposed to change the curvature of the lens, whether to see it clearly or to make it blur. So the image will be blurred as a result of inability of the ciliary muscle to be able to alter the shape of the what? The shape of the lens. And this is as a result of old age. When we are getting old, this is something that is very common to human beings, and this can be corrected by a kind of a glass, and this can be corrected by use of a kind of a specialized glass called bifocal glasses. We make use of bifocal glasses in correction of this presbyopia. As a particular point, we meant for the near object, while the other one we meant for the far distance object. That's why it is bifocal. One will be seeing the near object, the other one will be seeing the far object, and that is just that. And the fourth one now is what we call astigmatism. There is astigmatism, and this is a condition where the image will be blocked in the what? On the retina. That is as a result of when the light that enter, the light that enter into the retina does not evenly focus, does not evenly distributed on the what? On the retina. And this occurs as a result of curvature of the lens. When the curvature of the cornea and the curvature of the lens, it could be curvature of the lens or any of them. It could be either curvature of the lens or curvature of the cornea, or even the both. So as the image just enter, the light just enter into the what? Onto the retina. Instead of it to go straight onto the retina and fall on it, it will not be evenly focused. It will not be evenly distributed. So as a result of it, the image will just be blurred. It will be blurred on the what? And the vision will be blurred. And this can be simply corrected by a kind of a specialized glass called cylindrical glass. Cylindrical lens. We call it cylindrical lens. As it's the one that will help us to evenly distribute this the image on the what? On the retina. So this is the structure of human eye and the eye defect. And that is the end of the class. I still remain my name, Senator Idris Erinlakatabu. But before I go, don't forget to subscribe to Mr. Pure Biology. Thanks for listening.